Okay, hello everyone, and welcome for this first workshop session. Thank you for joining us today. And so today we are going to present you how we can produce double emission that will be compatible with FACS for cell sorting application. So before we get started, we will wait 2 minutes to let time for the latecomers to join us. I can see that people are still arriving. In the meantime, what I can say is that, so here we are making a workshop session. It's not as a, let's say, standard webinar. The idea of this session is to have good interactivity together. So the idea is to be as close as possible as a physical workshop. So you will see that we are going to present some slides, but there will be also a dedicated demonstration session. So we can show you how we can use our technologies to perform double emission, and also in order to you to more understand, so you will be able to understand a bit more the technology as well. Okay, so I think we are quite complete. So first of all, let me introduce you the team who will be presenting today. Youen Vitry and Adrien from Secoya Technologies. So they are both technology leads on encapsulation at Secoya, and they have expertise in emulsification, encapsulation inside single, double emission, complex emission as well. And from my side, Adam Meziane, I'm working at Fluigent, and I'm working as a project manager responsible for droplet and particle generation. Okay, so now let's move on to the presentation. A bit maybe of context before we go deeper in the presentation. So FACS, what does it mean? It means fluorescent activated cell sorting. And what is the interest of the FACS? So basically, if you are here, that means that you are aware about that, and maybe you already use FACS on a daily basis, let's say. But for those who are not aware, basically the FACS allows the analysis of the cellular behavior to understand the heterogeneity of cellular response, such as protein expression or based on enzymatic activity. So basically, it allows to analyze the cell secretion based on a specific, let's say, reaction. And the FACS, so it is a convenient tool to select among the complex population of cells those which show a specific fluorescent signal, and also indicating the production of a molecule of interest. For example, if you have an antigen antibody reaction, you will be able to observe with analyze with the FACS which cells are expressing this fluorescent signal. So as I said, the FACS is based actually on two things: light scattering first, and fluorescence. So the light scattering is based on a laser beam that will allow to, when the cells pass through the FACS, as we can see on the scheme, we will be able to analyze the size of the cells, and also the granularity, thanks to two detectors, forward scatter and side scatter. And also, as I said before, the fluorescent signal based on reaction, enzymatic reaction, for example, will allow to sort these specific cells based on the cell response. So we'll not go in detail to the FACS principle, but if you want, and if you have questions, we will go back during the Q&A session. Basically, so this is something which is widely used, but there is many limitation actually to this FACS. First of all, is that you will have, when you are making this kind of application using standard method, using FACS, you have one cell inside a big volume. So you have a single cell, let's say, expression, but in a bulk assay. So you do not have any compartmentalization. So how can we have better sensitivity, and how we can have actually a real single cell study using this FACS? So for that, there is a technique basically, which is called the single emission, that allows to compartmentalize the cells inside macroreactor. So the idea is to encapsulate cells inside single droplet. So inside droplet, basically. So you have an example here using microfluidic techniques. And by encapsulating into microreactor, you will be able to, after that, analyze the secretion of the cells. So what molecule will be secreted. And this molecule will be trapped inside this small reactor, so small microreactor. Using this small microreactor, you will be able to have a better analysis and better discrimination when you will use your FACS. So basically, when you are making these droplets, you will have aqueous solution with the cell droplets, which will be surrounding by an oil phase. Because to make emulsion, you will need two immiscible phases, water and oil, basically. So you will have the oil as a continuous phase that will protect the aqueous droplets, which will contain the FACS, which will contain the cells. Sorry. So this is an interesting technology. However, this is a technology which has limitation because the surrounding carrier phase is oil, and based on the technology of the FACS, the oil cannot be compatible with the FACS, because when you are using a FACS, actually you have a buffer, aqueous buffer, which is used to focus the cells so they can go one by one in front of the laser and also inside another. So that means you will have a biphasic solution if you are using an emulsion with oil. So how do we do to get the advantage of the encapsulation and also the advantage of the FACS? So for that, we need to go not on the single emission aspect, but go from the single emission to a double emission. So what does it mean? Basically, a double emission here will be to have a droplet inside the droplet. So inside, you will have a core droplet. For example, as you can see in green, in yellow-green, let's say, you will have your cells with your aqueous solution. It could be PBS or medium, for example. But surrounding, you will have a shell, thin shell that will be the oil. And as a continuous phase, then you will have aqueous solution. So you will have, let's say, an emulsion that will contain water in oil in water or in aqueous solution. So this is totally compatible with FACS because it's kind of having the cell suspension inside aqueous solution. So basically, as you can see on the scheme here, there is a small more explanation. So inside you will have the core phase that will be aqueous solution with cells, surrounding by the shell phase, which will be the oil, and surrounding that will be aqueous solution. So kind of the same setup when you are using with your cells. So now we know how to have the benefit from the compartmentalization and also be able to study at single cell level and also be compatible with FACS. But actually, there is many challenges to be totally compatible with FACS. To make a system which is compatible with the FACS, we need to really control the size of the double emission. The size of the double emission is an important parameter because in order to be compatible with FACS, you will need to have a double emission that will be smaller than the capillaries present in the FACS. And also, as you can see, in the FACS, you are passing through a nozzle. So if the droplet, the double emission is bigger than the FACS nozzle, you might destroy your double emission and you will not have something which is stable. And you will not be able to have your result at the end of your single cell study, as you can see on the scheme. Another important parameter is the monodispersity. You will need to have double emission with high monodispersity to have always the same sensitivity, so the better sensitivity. For after that, after analyzing with the FACS, have a better discrimination and be able to detect which cells are present, are expressing something, and the other which are not expressing. Another important parameter is the oil shell. You need to have a thin oil shell in order to avoid sedimentation of the double emission inside the capillaries inside the FACS. and also to have a good analysis. And at the end, you will also, of course, you will need to have good reproducibility to be able to perform your double emission in an easy way that will not be time consuming. So you can make and focus on the FACS analysis and on the result and not on the encapsulation system. So there is many challenges, and to address these challenges, there is different technologies that can make encapsulation inside double emission, but they are all presenting advantages and limitations. So in terms of technology, we have basically two kinds of technologies. The first one is basically the let's say we are calling in batch method. So this is mixing of two different solutions in the batch method, or using a vortex solution. And the other will be microfluidic techniques. So maybe Youen, could you go to the next slide, please? Yeah, perfect. So there is, as I said, two techniques: mixing in bulk or vortex. For example, the idea is to have, let's say, a reservoir that you will fill with two immiscible phase, and you will mix it together. You can use directly a vortex, or you can use mechanical forces, the stirrer, for example, and you will be able to generate a single emission. And then by repeating 2 times this step, you will be able to generate double emission. So this is something which is easy to handle, which is widely used. You have a high, let's say, robustness, because each experiment is quite easy to handle, and at the end you will have some results. However, using these techniques, you have really low control on the double emission size, on the droplet size, and on double emission side. So that means that you will have you will have polydispersed double emission population, and that could impact at the end your analysis in your FACS. The other method that could appear to solve the previous limitation is using standard microfluidic chips. So here you have a specific design with 2 identical design, but this design will be coded differently. One will be hydrophilic and the other will be hydrophobic, and that will allow to generate water in oil in water double emission, or the opposite, for example. So this is something which is a technique that is better in terms of monodispersity. When you manage to generate double emission, you have better monodispersity compared to the bulk or the batch method. However, it's something which is really hard to handle, and you have a low robustness because the channels are coated, and every coating has a lifetime. And so that means that it can run from some experiment, but not at the end. You will need to replace the chips, and it could be time consuming to set up the parameter. So this, in regard of all this limitation of this technology, the idea of this workshop is to present you a technology based on the development of a device by Secoya, and the combination of our pressure controller, pressure-based flow controller, and the emulsification technology that Secoya has developed to propose you a technology that allows to encapsulate biological sample inside double emission in an easy and more robust way. So now maybe Adrien, I will give you the lead.
Yes, thank you, Adam. Hello, everyone. I'm Adrien 𝗿𝗏𝘂𝘃, technology lead at Secoya Technologies. I'm very pleased to introduce you to our emulsification technology. I'm going now to give you just a brief general overview of the technology, and then I will let the floor to my colleague Youen about the FACS application and the live demo of our equipment. So at Secoya, we developed a device called the Raydrop for the production of highly controlled single and double emulsion. It is based on the alignment of 2 glass capillaries into a metallic chamber. This metallic chamber. 2 inserts supporting the capillaries are introduced on the lateral side of this chamber. The injection insert is connected to the droplet face, and it is equipped with a 3D printed nozzle attached onto the tip of the glass capillary. The collection insert, this one, support the collection capillary and is the only way out for the fluids, the droplet phase and the continuous phase filling the main chamber. At the entrance of the output capillary, the continuous phase squeezes the droplet phase flowing out of the nozzle and resulting in the formation of perfectly identical droplets. In the double emulsion case, the nozzle is fed with two fluids, the core phase and the shell phase, with the shell surrounding the core at the tip of the nozzle, resulting in the formation of double emulsion. And with the Raydrop, you can produce double emulsion with water in water, or oil in water in oil, or even with partially miscible fluids using the same system. Why? Because the droplet is never in contact with the wall of the capillary, and so it doesn't need the application of specific surface coatings in this system. And as you will see in the live demo, you can adapt the shell thickness by playing with the flow rates, but while always keeping a size dispersion lower than 2%. Using the Raydrop, you have access to a wide range of droplets and double emulsion adapted to numerous applications like API encapsulation in aqueous or organic solution. But actually, practically all the single and double emulsion processes described in the microfluidic literature, usually in PDMS or glass chip, can be done with this system. Okay, but as you maybe know, if you work with microfluidic, a lab on a chip can quickly become a mess on a lab due to the tubing spaghetti and equipment needed to operate the device. And to overcome this, we developed at Secoya an integrative platform that you will see in the live demo. And this platform includes all the tubing and equipment needed to control and observe the emulsion production, and of course the single or double emulsion Raydrop that is placed just over there between the camera and the light. This platform allows non-experts to quickly master the production of droplets so that he can focus on what matters for this research instead of consuming time trying to seal a PDMS chip or fighting with wetting issues or other time-consuming stuff. Okay, that's it for my general introduction. Now you are going to discover how we can use the Raydrop and the platform for FACS application with Youen.
Thank you, Adrien. So when we designed the double emulsion Raydrop, we aimed at the same specifications as the original Raydrop, meaning 100 micrometer double emulsion. With this geometry, we can produce double emulsion between 90 micron and 140 micron. Then we developed a new design to produce larger double emulsion. To do so, we simply enlarge our geometry, and we are now able to produce double emulsion up to 400 micrometer. This design is perfectly fitted for fast screening of new formulation. However, producing smaller double emulsion is more tricky. You can't simply scale down the geometry because the system will be more prone to clogging. The idea here is to use a counter nozzle in front of the double emulsion nozzle that will locally reduce the geometry and thus reduce the size of double emulsion produced. Thanks to this design, we can now produce double emulsion down to 25 micrometer, which makes them compatible with FACS application. For instance, the picture at the bottom left is a zoom in of the above picture. Here you can see calibration beads used for FACS calibration being encapsulated in a double emulsion. Right here. It's a bit difficult to see it, but trust me, there is some particle in here. When you work with microfluidic, you want to avoid dust and particles in your system because they will clog your system. For instance, we usually filter our solution with 0.2 micrometer filters. However, cell solution cannot be filtered, and the best that can be used are 40 micrometer cell strainer. This will lead to particles up to 40 micrometer being present in your solution. Therefore, in such a case, it is best to use a slightly larger nozzle than our original design with a core nozzle of 30 micrometer. For instance, in the picture of the right, we use a 60 micrometer core nozzle with a 60 micrometer counter nozzle in order to encapsulate cells in 50 micrometer double emulsion. As a test case, we use our equipment to encapsulate human PBMCs in 50 micrometer double emulsion. The solution for the continuous shells and core-phase are quite classical for this type of application. So we use PBS with 𝗵𝗵𝗃𝗁𝗃5000 and 𝗙𝗃𝗃𝐹68 for the continuous phase. For the shell phase, we use 𝗙𝗺𝗃, which is 𝗊𝗄𝗃, so fluoridated oil with 2% biocompatible surfactant. And for the core phase, we had human PBMCs stained with cell trace violet in gross medium. And on the right part of the screen, you can see a picture where you see a lot of double emulsion, which are monodisperse and around 50 micrometer in diameter. In some of them, you can see a violet dot, so here, which correspond to the fluorescent signal emitted by the cell. Another thing to take into account when you work with biological material is that you generally do not have a huge amount of solution. On the other hand, working with a Raydrop requires at least 0.5 ml of core phase solution to prime and stabilize the system. This quantity of solution can sometimes represent more than the amount of biological material available. Therefore, in order to be able to encapsulate low amount of liquids, we add an injection loop to the core flow path of our Raydrop platform. You can see it here. The idea here is to prime your system with regular solutions such as gross media or medium or PBS. Load your sample in the loop, in the injection loop here. And then when switching the valve, the loop will integrate the flow path of the core phase and will be encapsulated in the double emulsion. Thanks to this injection loop, you can work with small amount of solution without any loss. It can also be used to encapsulate different samples one at a time. Another use case for the Raydrop platform that we depict here. So in some application, you require to mix a reactant with the cells prior their encapsulation. For instance, this strategy can be used to look at single cell phenotyping. This can easily be done with the Raydrop platform. To do so, you simply have to add an extra fluid line that you can see here. You then use a T-junction to mix both core phase at the core inlet of the Raydrop. In such a case, the time between the control mixing and the encapsulation is below 20 seconds. We use this system to encapsulate cells with fluorescent particles. In the picture on the bottom left, you can see the nanoparticles being encapsulated inside the double emulsion, right here. In the picture on the right, you can see the double emulsion that contains the fluorescent nanoparticles. And we can now continue this workshop with a live demo of our equipment. So, before starting with the demonstration, I will present you the Raydrop platform from the top view, I would say. So I will first start by stopping to share screen and going with the webcam. Adam, can you pin us for everybody?
Yeah.
Okay, perfect. So it will be more smooth for hand gesture and video. So here you have the Raydrop platform. On the left part, you have the 3D installation, I would say, with the pressure controller at the bottom here. You will have then the reservoir, so continuous phase reservoir, shell phase reservoir, and core phase reservoir. You then have filters in the back. You will have then flow meter here. A set of valves that are really helpful to start and prime the system, so you can switch from the reservoir to the waste or to the Raydrop for each phase. And it's really important to use this valve to start the Raydrop encapsulation process. Then on the right part of the video here, so you have the injection loop right here. You have the Raydrop that is presented here, and then you have a small microscopic setup here that allow to see the emulsion generation and be sure that everything is working well. as you can see in the here, so as you can see, there is a couple of oil droplets that are stuck to the Raydrop nozzle, but it doesn't change the operation and the way it behaves. So no worries for that. At the top of your screen, you have the three pressure controller and the three flow rate. So for the continuous, the shell, and the core phase, and the same for the flow rate, continuous, shell, and core. And on the right, there is the injection loop. So when we start with the Raydrop, we will start by applying a pressure on the continuous phase. In the present case, we'll work in flow rate mode. So we impose flow rate, and the software will adapt the pressure on the continuous phase. We will then add some pressure on the shell phase. We'll switch the valve from the reservoir to the Raydrop. We will increase the pressure in order to flush all the remaining water from the continuous phase in the shell part to produce a jetting like this. When everything is stable and clean, we can reduce the pressure in order to reach around 10 microliters per minute. We will now do the same for the core phase, and you can already see the production of double emulsion. Of course, it's not perfect at the moment because you have to adapt the flow rate of core and shell phase.
Okay, and you can also see small droplets, that is satellite droplets, but actually this small droplets it's not possible to avoid this formation. It is due to the fluidic operation of the system, the dreaming mode. But it's not a problem for the FACS application because it will be analyzed as the noise signal in the FACS system. So what you see on the screen is double emulsion of water in oil, and the small droplets are just satellite droplets formed at each double emulsion droplet formation.
And now on the screen on the right, you can see the collection of the double emulsion, and you can see that they are all monodispersed. If you want to see a better view of the formation of the double emulsion, we have two videos. So this first one is with the contour nozzle. So as you can see, it's really difficult to have a good image because the process is too fast, and the lighting conditions due to the contour nozzle are not really easy. But we can see at the bottom a video that is really slowed down with a large configuration. And it's really easier to understand the formation, the process of creation of a double emulsion.
Okay, and at the same time, we have some questions. Do you offer an option with glass nozzle instead of the 3D printed nozzle? Yes, actually, Secoya developed also a nozzle in glass. So for better compatibility, for example, if you're using solvents, specific solvents that are not compatible with the 3D printed nozzle, it's possible to use glass nozzle as well. The second question is, can you take primary emulsion, water and oil, and make it into a double emulsion, water and oil and water? Yes, it is possible. However, for that, I would not then recommend to use the Raydrop double emulsion because it's made to make double emulsion basically. So you can do it using a single emulsion Raydrop, but you do not need the double emulsion is made. The Raydrop double emulsion is made to make double emulsion directly in one step. So that will be a waste of time to make your primary emulsion at the beginning and then inject it into the Raydrop.
There is a question about the satellite droplets. Are they always present? Actually, no. It depends on the fluidic regime. But in the regime we use to form this very small droplet for the FACS application and the fluids that we use, so this oil and the core phase and the continuous phase, yes, I would say that in this case the droplets are not avoidable, but it is not always the case. In the video we show you precedently, there were no satellite droplets. So it depends on the fluids and on the fluidic regime. But in the FACS application setup, yes, you will always get this satellite. But as I said, that is not an issue with for the FACS analysis.
Yes, exactly. This is something that we have to think about, because during your analysis, you will see the satellite as, if you are using the FACS with your cells, you see some debris sometimes, some debris. And the debris can be sorted as well. So that will be the same for the satellites. So for this configuration, yes, this is very satellites, but this is not a problem.
For instance, here, what do produce the satellite, can kind of do produce them, is the addition to the continuous phase of 𝗵𝗃. It's really the one that is creating this behavior, actually.
Another question: what is the ratio of the double emission droplet and the unsuccessful single droplets? If it's the satellites,
actually, it's kind of combined with the previous question.
Yeah,
there is kind of no numbers, I would say, directly available. So you have to test it with your own formulation. But it will really depend on the formulation that you use. In some case, as we show in the video just before, you will have almost no satellite droplet, I would say. Just maybe a small one, really small one, that is due to the regime condition, but these are less present.
Yes, but I think that the question was not about a satellite droplet. It was about single droplets and double droplets of the same size. But in this case, it's 100%. You will always get one double emulsion droplets and one satellite, but it's 100% of a double emulsion. The single emulsion that you get at the output is just the satellite. There is no single emulsion droplet of the same size of a double emulsion.
So you can see here, so the double emulsion is really the big one, and the satellite droplets are behind and are really small.
Yeah, and there is sometimes more than one satellite droplet per double emulsion droplet. So that's why you have the impression that there is more single small droplets than double emulsion, because the first satellite will produce a second satellite actually.
Okay, there is another question. Can you please specify the stability details of the emulsion formed? I mean, to what time the formed emulsion with Raydrop will be stable? It really depends on the application actually, and for the stability of the Raydrop of the double emulsion is not, let's say, it's not more on the droplet generation process. It's more on the formulation and the surfactant that you are going to use to keep it stable. So basically, here you can generate it and have double emission, but to keep it stable, you will need to have a strong surfactant. For example, application in PCR or when you need to make incubation, depending on the surfactant that you are using, you will need to have up to days of incubation. So this really depends on the surfactant that you are using. We have tried with the 𝗙𝗺𝘂𝗃, which is a surfactant that we are proposing as Fluigent, and these were really stable during days actually, and we tested it was stable. There is another question. Is it possible to purchase microfluidic cells, nozzles, separately with other setup parts? If it's only the nozzle for the Raydrop, is it possible? Yes, to purchase it separately. But yeah, it's made to work with the Raydrop. So if you already have a Raydrop and you want to change the nozzle, is it possible? It is exchangeable. So you can buy another nozzle, let's say, and you can exchange. But so yes, it is possible.
But regarding the use of this configuration to produce double emulsion, I would never recommend to buy the Raydrop by itself and buy other equipment and try to do a mix in your own lab, because it's kind of tricky condition to produce such a small droplets, and the Raydrop platform helps really a lot to start to prime the system. The valves seems to be something that is really simple, but it's the philosophy of the system helps a lot, and the integration of the equipment really helps a lot to produce this type of emulsion.
Yes, and the system is made of 3 parts, as I said before. So there is this main chamber and the 2 inserts, but the main chamber is always the same. So you can change the insert to go from the single emulsion to double emulsion if you want, but you can also change the nozzle if you want a bigger nozzle or smaller nozzle to achieve smaller droplets. It's possible to just change one or the other parts to adapt the geometry to your needs. That is of course possible. That is a quite versatile system that can be adapted to your needs. There is a question about the compatibility of our system with drug encapsulation in particle production. Or it is a 2-component based system only. Yes, we can produce double emulsion. That is the maximum. There is no triple emulsion system or more composition. So we can make double emulsion, but to my knowledge, it's enough to make drug encapsulation. Can you show the slide with what we can do with the Raydrop? Yes, no, this one. Yes, this one. On the slide, you see some example of what we did with the Raydrop single and double emulsion. So, by example, in simple emulsion, we can produce droplets of, by example, alginate or 𝗏𝗏𝗏𝗏, but also of 𝗙𝗏𝗂𝗏𝗏. So we can produce particles of 𝗙𝗏𝗏𝗏, and in this particle, we can encapsulate proteins or other compounds. And in double emulsion, what we can do is to produce microcapsules. And by microcapsule, I mean a solid shell, by example, with ketosane shell, a 𝗏𝗏𝗏𝗏𝗏 shell, or 𝗙𝗏𝗏𝗏𝗏 shell. And inside this solid shell, you have your API, your drug, in solution or in suspension in aqueous or organic phase. So yes, with our system, we can encapsulate API in microparticles using simple emulsion. So your API is in solid beads, or in double emulsion, your API is in solid microcapsule.
There is a question regarding the oil thickness. The thickness of the oil layer. So if we go back at the presentation, yes, just this slide. So actually, you have there is no fixed shell thickness. The shell thickness can be adapted based on the flow rate combination that you use, but there will be a regime where it is stable and not stable, because if you have not enough core phase, then you will produce only 1 double emulsion every 2 droplets, so it will not be really practical. And if there is a too high core phase flow rate, so an unbalanced flow rate between the core and the shell phase, you will break the system, so you will no more be able to produce double emulsion, and you will have to reduce the core phase flow rate to start to make double emulsion again. So it's a bit difficult to answer to this question, but in this application, the shell thickness is between 1-2 micron at the minimum case, but it's really tricky to produce, and 5 micrometer, I would say. But again, it will depend on the formulation and the oil that you use and the whole system.
The next question is about the stability of the double emulsion, water in oil in water. How long can they incubate it? I would say that the stability of the double emulsion is not dependent of the Raydrop. It's dependent on the formulation. Depending on your formulation, the double emulsion will be stable for a long time or not. And actually, the main factor is the presence of surfactant to avoid coalescence in your system. So that is very formulation dependent. There is no general answer to this question. But if you work on the formulation, you can achieve a very long time of incubation.
Yeah, if I can add maybe, so we have made some experiments more in single emulsion for encapsulation, for example, in microbiome, where we used the surfactant that we are proposing. And we needed to have encapsulation and then incubation for different days, up to 7 days, for example. And it works really well for this application. So again, it's really dependent on the surfactants for single and double emulsion.
The next question, so it's is about the minimal droplet size that we can made for FACS application. So I would say that it's more in general manner. The lower that we produced was 23.2 micrometer in mean diameter of double emulsion. We could go lower with for instance a counter nozzle of 30 micron, but then the system will be really tricky to master because we will have to work in a really clean environment. So for now, we stick to this minimum size, but if there is demand, we can still do it. It's doable.
Yeah, from our knowledge, when we were discussing, so below 30 micrometer for FACS was acceptable. After that, we never saw lower than 25 or 23 micrometer, but if it's a need, we can discuss.
The next question is about the typical shelf life of the nozzle. Because there is no surface modification of the nozzle, that is true. There is no application of any coating on the nozzle. We don't need it because there is a cylindrical geometry, so there is no contact between the droplets and the wall of the system. So because it doesn't need this surface modification, the shelf life can be very long and will actually be dependent of what you do with your nozzle. If you work properly, if there is no clogging with such a hard polymer, if you use the right solvent, so there is some solvent not compatible with this nozzle, I would say that the shelf life, the typical shelf life, could be 1 year. I'm working with one nozzle every day from 6 months, I would say, and there is no problem with this, because if you work with mild solvents compatible with the nozzle, so all the classical solvents are compatible, but there is some hard solvent like dichloromethane that are not compatible. In this case, you need to work with the glass nozzle. But yes, I would say typical shelf life is about 1 year. The next question is about the rate of droplet formation. It will depend on the size of the droplets. If you work with very small droplets, for the same flow rates of droplet phase, you will have a very high rate of droplet formation. In some experiments, we reach kilohertz of droplets of 30 microns diameter. But if you are working with bigger droplets with another configuration, with a big nozzle, typically the flow rate for the formation of droplet is about tenths of hertz. So the typical rate of formation is dependent of the size of the droplets, from tenths of hertz to kilohertz, depending on the size.
The next question is about if our system is industrialized. Yes, we are working on a parallel version of our technology, and we expect to have good results by the end of 2023.
But yes, I would add that this scaling up is a scaling up by parallelization. So the recipe, the process can be debugged using a single line system like this one, and then the recipe will be totally applicable in a larger scale equipment. That is the way we work. So the development that you can made with the single line system will be totally and directly applicable to the next equipment for large volume.
There is another question regarding FACS analyzing. So what is the ideal overall size of a droplet and ratio of all shell to inner phase that is compatible with FACS? Actually, it totally depends. So for the size of the double emission, it totally depends on the FACS that you are using and on the nozzle present on the FACS. There are nozzle of the FACS that have 85 micrometer. Other, they have 50 micrometer. So depending on that, you will not be able to generate 100 micrometer double emission and keep it stable during the FACS analyzing. So basically, it's based on that. And for the oil shell, actually, it's really important to manage to have a thin oil shell to be able first to avoid sedimentation of the double emission inside the FACS, and also to be able to have a good analysis based on the laser beam that you are going to send to the cells to analyze the cell size, the cell size, the double emission size, the granularity, sorry, and to have also a fluorescent signal. And there is another question which is kind of related. Can the thin oil film of double emission droplet cause some problems during FACS reading due to the index of refraction change, water or water? Yes, this is exactly the point. So the idea is to while keeping a stable double emission, avoid to have a too large shell thickness. So the smaller, the better, to have a good refraction and also to have a good analysis and good reading of the FACS and avoid sedimentation while keeping the double emission stable. So this is totally a balance. Okay, I don't see any more questions. So if you still have some question, ask it maybe now so we can answer it. Otherwise, so we are thanking you for joining, for attending this webinar, this workshop. I mean, we will send you email with the replay of this session next week. And also, we will send you also in this email the ability to book via Calendly session to book a meeting with us if you have specific, for example, question regarding your application. If you have other question that you didn't ask today, and we will be able to discuss in one-on-one meeting. So, no more question. Thank you, everyone, and have a good day.
Thank you.
Thank you. Bye.
Bye-bye.